So thanks for doing this panel discussion. And I have some prepared questions. And of course, I'll welcome questions from our audience. Please just raise your hands anytime if you want to ask anything. So I'll start with the first question, which I think is most important for this symposium, which is the AI rigor and validation. The question is, what does statistical rigor mean to each of you in the context of AI applications? Where do you see the biggest gaps between current AI practices in biomedicine and the standards you'd expect from traditional statistical inference? Maybe we'll start from Wei. Yeah, since you're sitting on the right. Yeah.
Well, I was still briefly because something. Okay, sorry. Something very simple, just neural network seems always good. The overfitting is really a concern. It happens like one example. Let's talk about the HE foundation model. Like we also work on this, and then we try to use HE image to predict gene expression. If you got those HE image, you randomly split them, and the testing data actually perform pretty good. But if you split the data by patient, right, this 50 patients are training, the other 50 are testing, the performance are lower. Because if you randomly split, there's some image tiles from the same patient from both training and testing. And it's very easy to so something easier, but the situation is much easier, you got much higher performance, and then people want to see it. Like you got good performance, right? So I'm going to stir that. I think that's a risk fitting. Yeah.
You stole my answer. I mean, I think that's true. Overfitting. So the general problem of leakage is a huge problem, and I think it's not well understood or acknowledged by many practicing machine learning researchers. I think the one challenge is that people don't necessarily distinguish between whether they're interested in a truly inductive versus a transductive setting. Are you training on data for which you don't have the labels, but you have available at training time? Or do you run inferences on data that wasn't available at training time? Right? A lot of these models are trained, in scenarios that aren't actually interesting. Like you train them to impute missing values from chromosomes that you didn't train on. Of course, the data for all the chromosomes was generated all in the same experiment, so it was all of it. It's all generated at once. And I think the way people write papers doesn't necessarily acknowledge, what the actual setting is for their train test split many times.
So I totally agree that the validation data set and the training data set, the differences between those two is a key issue. And I'm happy to report that in the review process, many reviewers are very keen on that point. So I'm hopeful that as we progress, it will be taken more and more seriously, and that will lead to solutions in the future.
Thank you.
Yeah. So an important issue was already raised. I think sort of more generally, the idea of generalizability of these models is really important. Yeah. Now, still no? Shouting, shout. So generalizability, as mentioned, I think that's a hugely important issue. The things that I've been thinking about when we sort of talk about it. So evaluation and sort of how do we evaluate whether these AI models are doing what they're expected to do in different problems and different settings. I think there's a whole statisticians in particular that sort of is happening in that space. One that I think is important is quantifying uncertainty. So Jeff's been doing a little in synthetic data. We've been doing some work with synthetic data. So when you use synthetic data and sort of the foundation models that were discussed, how do you evaluate the uncertainty that comes from inference that is done using synthetic data? It's a hugely important problem, and it's, I think, not quite addressed.
this conversation about sort of interpretation and explainability of these models that was also brought up, and I think that that's also a really important issue. How do we do inference with these models? Yes, Shapley values are great. I mean, they give us some sense of qualitative sense of what are factors that are important in making the prediction or sort of the decisions that these AI models are coming up with. But so that's not necessarily the scientific question that we were interested in to begin with. That's just an interpretation. And that connects with my last piece, that sort of the design or thinking behind sort of when we use AI. What's the scientific question that sort of we have when we use these AI tools for, and how do we do inference those scientific questions? I think that that's something that sort of is really important as well.
Lucas.
Thank you, Ali. So I actually agree with most of your point, and I would say I use a different word where I personally super concerned about is the trustworthy of AI. So I would say like in the two component of it, most important to me is first of all is the model evaluation, where I was what I mean is the model robustness. Like how can we make sure that the model you train once is actually predict. So we show the performance of that performance, but if we train it another time with a different seed, we'll get a different performance. Then this happens to all of my project. If you do not change any of the prep, any of the model architectures, but just to play with the seed, you can get total different model performance. And I don't know which one to trust, and I don't know which one to report. And also the second thing also related to trustworthy of AI is the interpretability. And in one of the projects I just presented today for pathology images, even though we showed some of the heat maps, show that the model is focused on which part of the tiles to predict the mutations, but actually there's no biological interpretation of it, why that tile is most important to that prediction. We did a little bit of contribution there saying, okay, we're looking at the most true positive happening in the cancer cells. But we're also looking at the microenvironment, since MSI is related to immunotherapy response. We're looking at the microenvironment of those tiles. There's no association between the known biomarkers in microenvironment feature space. So the model, like how the model had that great prediction with ARC AUC more than 90%. I don't know. So I think we should put more effort into interpretability of AI and also the model evaluation.
Thank you. And Jeff?
It's tough being last to say something unique. Or not last, like a Jonathan might be. He's even in a tougher spot than me. I would say that the main thing I think about is how much the models that we're going to be using in the future, how little we're going to be able to interpret them as they get bigger and bigger and more complex. And so to Lucas's point, it's important when you can to interpret them, but often you can't. And so then I think the thing that becomes really important is independent evaluation of the models. In other words, in every paper I review, somebody has come up with their own evaluation of how to evaluate their model. And surprisingly enough, their model looks amazing on the evaluation that they developed for their paper. What is, I think, more interesting is when they show that somebody else developed an evaluation and they tried on that evaluation and it worked pretty well. And so how do we as a field define, for the problems that are the most important, what are the independent evaluations that we want to do so that people can really tell if something's real or not when you can't interpret the model?
Thank you. And Jonathan?
Yes. Hello, everyone. And apologies, I can't be there in person. I'm actually at NIH this week, where I'm talking about using AI for health behavior change. So I have a complementary perspective to the panelists, and that is what the AI systems can do to promote behavior changes that prevent cancer, for whom, under what conditions. And so we're doing this with several projects at the Cancer Center for quitting smoking with native populations and with the general population. So the question isn't just, does it generate reasonable text, but what is the causal effect of this behavior change agent, which we create large language model chatbots, on cessation, on engagement, on long-term behavior change, and is that transferable across populations? So I think the biggest gap I see is that AI papers are often emphasizing predictive performance, whereas prevention science is asking a different question. Given a certain pre-specified outcome and a comparable condition, are you going to see AI-based behavior change agents actually produce long-term change that's replicable? So we have it's a different aspect that I think really would what I look at when I think of statistical rigor in AI applications.
Thank you. And thank you for our panelists' comments. I just want to add one thing. A lot of our discussion is surrounding prediction, but nowadays a lot of people use generative AI to generate fake data or synthetic data, like image generation or video generation, even though that may seem a little bit far away from biomedical research. But my take is that maybe we need different consideration or statistical rigor when we talk about generation versus prediction, right? If we say synthetic data looks real, how real should we be trusting it? That's another question. I'm just throwing the question here. All right, do we have any questions from the audience since we had a long discussion for the first question? Yes.
So I have two. One is for Jonathan. Jonathan, I think that's interesting your take. I mean, it does make me think about the predictive models in say treatment, like predicting outcomes and then predicting benefit from a treatment. Would you say that when you're talking about a preventive intervention and predicting whether it's going to be beneficial, that would it be analogous to basically you're predicting benefit instead of predicting outcome?
No, we're predicting outcome. It's just that given the model, when you compare it to other treatments that are available, is it going to outperform
I see.
the?
Okay.
That's what how we think of it. So in those terms, we need to look at utilization, we need to look at engagement, we need to track those things over time and be able to and look at what kinds of conversations people are having with these large language models to see if they're clinically useful conversations, those sorts of things.
The other point I wanted to ask the panel, you mentioned generalizability, and there's kind of a bar for generalizability that I see in published studies where, I mean, Lucas showed it. You basically train on a certain data set. You have to show that it performs similarly on a different data set. And it seems to me that that's a rather low bar. And I mean, what do you think about that? Is that something? And the new data set could be different quality. Lucas had that Neptune's data set, and I just wonder if we're missing something on what we're asking people to do to demonstrate validation and generalizability.
Yeah. Any volunteers?
If somebody has another answer, I'll just briefly say I think that it is a relatively low bar in the sense that you're not asking it to do a different task. You're usually asking it to do the same task on a different data set. I do think that that's a good bar to have things leap over because we have so many models that get developed in biomedical science that get developed on one data set and just don't even clear that low bar. And so I think, and part of that's data leakage and other things, I think. One thing I'm really interested in is the generalizability of especially generative algorithms we build. And that's because you think about, Anthropic's models or OpenAI's models are trained on Reddit, but can say something about biomedicine. It's sort of interesting to see what the generalizability of generative models are for tasks that are way beyond just reproducing a single prediction task. But I think my sense is that, again, going back to beating that drum of like, we would need to know first, what are we trying to get it to do? What are the evaluations we actually even want to do before we can ask it if it jumps over those bars?
Yeah, I totally agree that, I mean, it's important to be able to generalize to a new data set, but it's also important to keep in mind what the notion of a new data set is very domain specific, right? I mean, you can have some domains where it's very replicable and some where variability is very high between different data sets. So I think it would go back to two things. One is using established benchmarks. So like not inventing your own performance measure, but using the measures that other people have used and the data sets they've used is a good thing that we should encourage. And then also comparing to other existing methods that try to solve the same problem. So you're not just saying, I work well in an absolute sense, but I work well in a relative sense to other state-of-the-art techniques. I think those are probably more important than being able to say, yeah, I got new data and it worked well on that too.
Maybe I'll add just one bit to this, that I think just building on what Bill said, thinking about sort of metrics that are established metrics. I think it's also important to think, especially in clinical settings, those metrics are not necessarily the same. So AUC sort of, who cares in clinical setting? So we have to really think hard about sort of what is important in clinical setting. It's like numbers to treat, maybe sort of positive predictive values. So we have to think really hard about sort of the setting. I mean, if it's a clinical setting, especially, so that's another thing to consider.
Okay. Sure. Yeah.
I think that's a great point. It's really not about replicating a new data set. It's more about thinking about the intended clinical settings and then what kind of consequences or outcome you expect, and whether you can, whatever you develop can achieve that goal. So I think that the fundamental biostatistical common sense we've learned in the past before the AI era still holds here. You really need to think about the data set you used, for the validation of development, how relevant, clinical relevance that is. So I think the study design is also very important. You have to ask where your data set come from, and they need to be collected also in a rigorous way.
Thank you. So for the remaining time, we have 10 minutes. I would like to just go back to our theme of across scales and ask our panelists a question related to the scale of the studies they do. So I'll start with Bill and Wei, since they focus more on the molecular biology level. I want to ask you this question. How do you balance model complexity with interpretability in your research?
I like the small model that Bill talked about. Maybe I'll touch this question in a little bit different point of view. I was thinking is also the amount of training data, the type of training data you have. Like one example I can think about is single cell foundation models. They predict the knockout effect. Most of them, they are failed. They use a huge amount of data and a huge model, but I think they're just fundamentally not the right type of data and model, because you think about their large language model. A cell is not really a language. It's more like a word model, right? When you want to predict autonomous driving, like you need a video to train it. You cannot just use picture to train it. So in that sense, I guess it's the question, how strong is signal to noise ratio? What kind of data you have? Yeah, I'll stop.
Yeah, so I think this is, I don't know if I can turn that on too, but I will. It's always a fundamental question, right? It goes back, the beginning of machine learning, like the notion of, are you doing a generative model or a discriminative model? Do you want the parameters of your model to have interpretable semantics, or are you just interested in answering the question at hand as accurately as possible? And I think that is a fundamental trade-off. So that's sort of like, when you design statistical tests, you have to think about is the application that you're actually interested in and design your test for that. In this setting, I think you have to ask yourself, how important is it that my predictions are accurate, and how much am I willing to give up in terms of accuracy to get a more interpretable model? You can't, yes, you can always get interpretations, but the interpretation will be better if you make your model a little bit less complex, for example. But obvious, benefits to having a linear model, like you can understand it much better. And yes, there are, like we said, there's SHAP values and so on can give you to try to tease out from these huge models some story about what they're doing, but it's not as even that is sort of there's some the faith there that you're trusting that that's what the model's really up to. So, I guess to answer the question, it depends on the application domain, who my collaborators are, and what they care about. Are they interested in the predictions per se, or is that the way you can get the explanation? Right. Often it's, if we understand molecular biology, the predictions themselves don't matter. We want hypotheses from inside the model, and that can tell me, let's use a simpler model. So those are the kind of considerations.
All right, my next question is for Yui and Ali. And I see both of your research kind of in the bridging sense, in that you have worked on many different applications from molecular biology data to more like clinical data, because you focus a lot of methodologies. I want to ask you to just mention or point out one most important statistical challenge you see from your research in terms of what can statistical uncertainty quantification or bias correction do for the applications you worked on.
Yeah. I'll go first only because my thought is still on the previous question as well. So I was thinking when Bill was talking was that it seems to me there are two scenarios where in one scenario, what we want the model to do is to capture the human intelligence, like to do something that humans can do, humans can see. So in that case, oftentimes we may not run the model to completely replace a human, but more have a human in the loop. So in that case, it still goes back to the validation. The things may be drifting, but if there's a human in the loop, and the human may have a suspicion about something weird going on, so if then the model can explain itself, then that may be a check. So that's one scenario, and that may be the relatively easier scenario. The harder scenario is that where we want the model to do something humanly impossible. So there are claims like the model can do better than human. So I think that's much harder. So there are different implications in terms of interpretability, and validation in that. So sorry.
Yeah, that's
Yeah, maybe I'll touch on the other side of this. I agree with the comments about interpretability and sort of what's the goal really of the analysis and of the scientific endeavor. But I'll touch on sort of the uncertainty quantification. So Lucas talked about very sort of important point that sort of if you retrain using different seeds, you get different answers. But that's only one source of variability in sort of the results that we get. If we think about sort of generating data, synthetic data, there's also sort of the uncertainty with the generations. So if you train it, the same model will give you different, actually sort of synthetic data. So that's two sources. And then there's a third source, the original data that we had sampled to then build this model from. So they're actually, what we've been thinking about is that there are three sources of variability that you have to account for in order to do valid inference. On top of these three sources of variability, there's also bias that comes with potentially using synthetic data that we have to adjust for. So developing approaches that would allow us to adjust for that bias, but also sort of account for these sources of variability when we use generative models is a really important statistical problem. It's difficult. At least one source of these variabilities we can't really quite get around because in some settings, I mean, for example, with synthetic data, if we're using them in data privacy settings or in even federated settings, we might not be able to get around that sort of first source of variability. So there are some complications, there are limits in what we could do, but then trying to figure out systematic approaches for at least quantifying as much of that uncertainty as possible and adjusting as much as that bias that is possible. That's something important.
Thank you. Yeah. All right, my next question is for Lucas and Jonathan. Lucas, your focus is on clinical data, and Jonathan is more on population data. And you talk about a lot of positive or successful examples in using machine learning and AI. I want you to maybe go back to the negative example to see. I'm very curious to see, because the statisticians often talk about uncertainty, bias correction, right? So these all sound kind of like negative words. Would you say there's a room for us to turn this negativity into positivity?
I'm sorry. So what negativity you're
Because I mean, uncertainty quantification, bias correction, these all sound negative, right? But if we can show that by doing so, we can find more reliable discoveries, we can lead to new discoveries, then people will be more convinced instead of just telling people, oh, you're wrong. Do you have any examples from your research?
I feel bias correction or uncertainty quantification, that to me is not this two phrase is not negative, but maybe bias or that itself is negative. I do not have a example, but in my previous research in EHR, dealing like training machine learning deep learning model on EHR to predict acute kidney injury. Those where we actually have a lot of bias on we actually have model performance on differently in Hispanic patient or in patient in color. So that there at that time I was purely in the computer science program, and I don't talk to statisticians. I don't know there is uncertainty complication, there's bias or that. So then after I joined Fred Hutch, actually I noticed that there is a lot of uncertainty in the data set, also bias to correct. And I feel like I just learned this recently for the last 2 years. I think we need help from the statisticians. For example, like how can we use statistical model to quantify the uncertainty in your input X, in your predict Y, and also the parameter estimator. So that's something that in my training I never learned how to do it. So that's where I feel like we really need your statisticians' help.
Thank you. And Jonathan, do you have anything to add?
Yeah, I think they enable discovery rather than replace theory-driven discovery. You know, in digital AI research that I'm using for behavior change, uncertainty and bias, I don't see them as like technical afterthoughts. They're part of the phenomenon. People disengage. The model can give inappropriate answers. People can respond in ways the model didn't expect. So when you're looking at AI in its application to human behavior change, that's going to be that uncertainty is par for the course. And I think what we have to do is be able to understand those uncertainties and know where these models are failing and for whom. So you can look at how are you going to improve them, and so to me that's opportunities for new discoveries, not as problems to avoid or see as negativities.
Okay, thank you so much. My final question is for Jeff. Since I know your research has spanned molecular data and now you're working on more clinical translational research, what is your take on the possible integration across scales and how can AI help us with that?
I would say a lot of folks here are doing that across scales in various ways. My feeling is that as we go forward, the multimodality of bringing multiple different data types together to try to answer questions is only going to increase. Sort of as the cost of generating data goes down, whether it's synthetic or old-fashioned in the lab, I think we're going to see more and more different multimodal data types about everybody we collect. Jonathan's experiments collect mountains of new kinds of data about behavior that we never collected before. And so I think we're going to continue to see that expansion in all areas. What I think is exciting about that, both as statisticians and as scientists generally, is that gives lots of opportunities for us to think about what the next generation of statistical problems are. I mean, there was, when I was just getting started, FDR was the big thing that everybody was talking about, and that was what my advisor worked on, and I've worked on stuff in that space. I think the next generation of problems are coming, whether they're synthetic data problems, multimodal integration problems, uncertainty quantification problems. And so it's kind of an exciting time to think about what's the next generation of statistical development that's going to happen over the next few years, which I think is going to be explosive and fun.